This video will cover the topic of meiosis. At a broad level, meiosis and mitosis are quite similar. Cells that undergo meiosis progress through the cell cycle steps, and in meiosis, individual steps have the same names as those in mitosis. However, a key difference is that there are two phases of meiosis, each with the five steps that we previously discussed in meiosis. Meiosis 1 is where the main differences in meiosis and mitosis occur. In this step, we separate homologous chromosomes, resulting in haploid cells by the end of meiosis 1. Meiosis 2, on the other hand, looks much the same as mitosis, with the exception that it is done on haploid cells rather than diploid cells, and results in four haploid daughter cells that are not identical. So with that in mind, let's back up to prophase 1 and see all the places that mitosis and meiosis differ. Prophase of meiosis 1 is subdivided into five stages shown here. The main thing that happens here is homologous chromosomes are condensed, paired up, aligned, and stitched together. When aligned, these homologous chromosomes often exchange pieces of themselves in a process called crossing over. Crossing over is the physical exchange of chromosome pieces during the Pachytene stage. This actually happens reasonably often, and we can use the frequency in genes that cross over to understand their relative positions on the chromosomes. At the molecular level, this process of crossing over forms a chiasma as the strands of DNA from homologs interact and eventually separate from the parent chromosome. What this process looks like at the molecular level is a subject for another video, though. Our final stages in prophase of meiosis 1 involve loosening the synaptonemal complex, which has stitched the chromosomes together up to now, and the dissolution of the nuclear membrane. Now that we have made it through prophase, let's move on to the later steps. Notice that in prometaphase, our homologous pairs of chromosomes are traveling together, and that in metaphase, we see the paired alignment along the metaphase plate. This is very different from what we observed in mitosis with the single file alignment. During anaphase, our homologous chromosomes separate, and by the end of telophase and cytokinesis are located in two separate cells. These cells are genetically distinct because only one copy of each homologous chromosome is found in each of the cells. So either the homolog you receive from your mom or dad is located in a new daughter cell, but none of our cells contain both copies of a single homolog, at least as when things go according to plan. Meiosis 2 is really just a repeat of mitosis, except for, as stated previously, the starting cells are haploid rather than diploid. This stage separates sister chromatids, resulting in four total daughter cells. Some key differences in mitosis and meiosis. Two diploid cells are formed in mitosis versus four haploid cells formed in meiosis. Mitosis produces genetically identical cells, whereas meiosis produces genetically distinct cells. The cells at the end of meiosis contain only one homologous chromosome of each pair. The specific homologs that are contained in daughter cells can be from a large number of combinations, and we will dig into that in a separate lecture.